Right, let's get started. So thanks everyone for joining us today for our LIPIDMAPS webinar, which will be presented by our guest speaker, Professor Pieter Dorrestein, entitled Emerging Big Data Era in Metabolomics, Lipidomics, Discovering New Molecules at the Repository Stage. It's going to be around 45 minutes, allowing 10 minutes at the end for questions. Today's webinar is being sponsored by Avanti Polar Lipids. We're grateful for their ongoing support, and I'd now like to introduce Pieter. So Professor Pieter Dorrestein is the Director of the Collaborative Mass Spectrometry Innovation Centre and the Co-Director at the Institute for Metabolomics Medicine at UCSD. He is originally from the Netherlands. He did his BA in chemistry in Northern Arizona University, followed by a PhD in chemical biology in Cornell with Neil Kelleher. He held an NRSA fellowship in bioanalytical chemistry at the University of Illinois. He works at the Skaggs School of Pharmacy and Pharmaceutical Sciences at UCSD, where he uses mass spectrometry to eavesdrop on the molecular conversations between microbes and their world. That's a very elegant way of putting it, Pieter. Pieter's work aims to develop new mass spectrometry methods to understand the chemistry of microbes, microbiome, and their ecological night. In particular, he specializes defining spatial distribution of natural products in 2D, 3D, and real time. And in 2016, he was profiled by Nature as the man who can map the chemicals all over your body. And I do remember that article back then. I know this is a number of years ago, so really, really fascinating. And so we're going to hear today about his work in the lipid field, and his particular areas of recent research directions include capturing mass spectrometry knowledge to understand the microbiome, non-invasive drug metabolism monitoring, informatics of metabolomics, which includes lipids, microbe immune cell, microbe host, stem cell, cancer cell interactions, and disease versus non-disease model organisms. So one of the things I think Pieter's going to be talking about today, which we had a brief chat about before the talk, was a recent paper that he had in Nature called Reverse Metabolomics for the Discovery of Chemical Structures for Humans. And this is a really interesting paper to us, particularly on lipid maps as well, where we capture structures into databases for users to find and discover. And in that paper, he took a computational approach to predict and then find new lipids from several classes in biological samples. So a repository of N-acyl amides, bile acid amides, and FAFAs was generated. And from that, we've already added a number of structures to one of our databases. And I was chatting before this talk with Pieter about how we work out what else to add to others, because I think there's a lot of scope for lipids to be added to several databases that we have at LIPIDMAPS here. So and this is a really exciting field that I think is being opened up right now to a large extent by Pieter's work. He's going to talk to us more about this today. And I'm really delighted to hand over now to Pieter Dorrestein for his talk.
Yeah, thank you. And also thank you for having me here. I'm going to go, really one of the key things that I'm going to try to do is cover some of the key tools that we've recently introduced. And I will highlight that with the bile acid as an example, but you can envision how a lot of these tools and resources that we're creating can be extended to other classes of lipids. I'm not a lipid expert. I accidentally often stumble across them because they are important in our studies. But the tools that I'm going to describe are really sort of reverse metabolomics of what that process looks like, microbe mass, and then a mass QL query. And these are the three key papers. And then one thing that you will notice is that there are a lot of authors on these papers. And I will not individually highlight each of these authors, but the work that I'm going to describe could not be done without all of these different authors. So they really contribute key parts to all these different projects that I'm going to highlight today. Also, before I go on, I'm a scientific co-founder and advisor. These are my disclosures of these different companies shown here. Fundamentally, in my laboratory, what I'm interested in is really the understanding of the kind of molecules that microbes make and their functional roles that they play. Or we're interested in the chemical environments that allow different microbial communities to thrive. So those are the two complementing things that we try to study in the context of my laboratory. But through that, we also build community resources. My laboratory focuses on untargeted mass spectrometry, and I think many of you are familiar with what untargeted mass spectrometry looks like. And that you collect a precursor scan, and then you get fragmentation data out of it. And this could be data dependent or data independent, but we tend to focus on data dependent methods. And once you have this data, there are actually 1,001 ways to analyze this data to inform biology. One of those would be trying to match against reference libraries to create annotations. And so if we think of a given untargeted metabolomics project, you can think of this as a dark sphere, so to speak. And this is all the data that is collected within a given project. And when you try to match against reference libraries, so in 2016, if you try to match against reference libraries, public reference libraries, I should say, you had about 1.8% annotation rate. And this has grown in 2024 to an average annotation rate of 14%. It still means that 86% is unannotated. But now if you go and look at those annotations that you do get, and you do Google searches in terms of trying to figure out what these molecules are, and you try to identify which ones are made by microbes, you'll see that a very small percentage, usually less than 1% of those 14%, ends up being able to be annotated to belong to microbes. And so if you contrast this to, let's say, metagenomic sequencing, so if you take a fecal sample and you do metagenomic sequencing and you look at all the genes that are present, you'll find that the majority of genes that are present belong to microbes, and a small proportion of it belongs to the human genome. And so there's a stark contrast between what we can call in terms of microbial metabolism versus what the genetic capacity is capable of likely producing. And so really, if we want to ultimately mechanistically understand the microbiome, we have to have all the puzzle pieces. So what are all the microbial molecules that exist? And so we spend a lot of time on this. And so we ask often very fundamental questions. So in this case, how does the microbiome really impact the chemistry of the host? And so how do you study this? Well, we took mice that were raised without microbes, so germ-free mice, and we then compared this to mice colonized with a normal microbial community, generally called specific pathogen-free or SPF mice. But you just have to think of these as these are raised with a normal, healthy microbial community. So in this case, we dissected 29 different organs into 100 different pieces and performed LC-MS analysis on this. So we had about 2,000 LC-MS runs, resulting in about 2 million MS-MS spectra. And so now the question is, how do we visualize this information? And so in 2012, we introduced something called molecular networking. And the way to think about molecular networking is if you have a series of molecules in a sample, you collect MS-MS spectra on them, so the ion forms of them. What we then do is we merge all identical spectra just to reduce the computational burden downstream. And then we represent these spectra often as nodes. And we can do something called a spectral alignment, where we take the precursor mass in account, and then we look for these ions of those precursor mass differences to enable doing spectral alignment. And then we build a network out of it, where you have a node that's represented by a specific molecule. And then if you have a delta mass of 147, it would correspond likely to a phenylalanine. But if this is a 14 dalton, it would be a methyl group. If this is 26, it would be a CHCH. If this was a 15.995, it would be an oxygen. And so this is what we did with the mouse data set. And in this case, we also color-coded based on the metadata. So if they're only observed in germ-free mice, they're blue. If they're only observed in SPF mice, they're green. And if they're shared between the two, they're in purple. And what you can see is that there are whole clusters that are present in mice that are colonized with a normal microbial community. And there are whole clusters that are present only in germ-free mice. And so this is really a visual picture of all the molecules or all the MS-MS spectra that we obtained in this particular data set. But we started, so this allowed us to sort of do node counting and say, how much does the microbiome really impact the chemistry of the host? And in certain regions, like the digestive tract, as much as 70% of the chemistry is different. Whereas if you go to distant organs, such as the brain, still there's a lot of chemistry that is different when you have a microbial community or not, because even the brain has as much as 20% of the chemistry that is different. This is not taking into account quantities of molecules. No, this is presence and absence of specific molecules. So that was really remarkable. One of those clusters that became very interesting was this cluster right here. And so we had a reference library match to glycolic acids, and you can see the MSMS spectrum here. And this was observed in both germ-free mice and SPF mice. But we were really interested in nodes in molecules that were really only produced by the microbes. And so we were looking for these nodes that were only observed in SPF mice. And what you can see is that based on MSMS spectral alignment, we can see that this node is related to this node over here, and that the delta mass is 90 Dalton. Now, originally I was trained as a proteomics person, and I recognized that this mass was a substitution of a glycine to a phenylalanine. And then we have a 15.996 Dalton difference between these two. That's an oxygen. And indeed, our predictions were that these had a phenylalanine loaded and a tyrosine loaded onto a trihydroxylated bile acids, because when you do MSMS analysis like this, it's really hard to differentiate regiochemistry and stereochemistry. And so because these studies were done in mice, generally people talk about mice having different bile acid panels to, let's say, humans. And the kind of bile acids, trihydroxylated bile acids that are found in mice are muric acids and not generally cholic acids. And so we predicted potentially these could be muric acid and not cholic acid. However, when we synthesized the muric acid and the cholic acid and did retention time analysis, it matched up to cholic acid conjugates and not the muric acid conjugates. And so to make a long story short, we were able to show that microbes, when you pre-incubate bile acids with them, are able to produce these in culture. And we just kept discovering new ones. So in this mass paper, we found serine, cholic acid in viral infections. We found the phenylalanine deoxycholic acid in a human study of non-alcoholic fatty liver disease. So these are twin study. In this chemical proportionality paper, we found the alanine cholic acid. In this prediction paper, so to speak, we verified the phenylalanine chenodeoxycholic acid and the tryptophan chenodeoxycholic acid. But we kept finding more and more and more. And so this is when Emily Gentry, who's now a faculty member at Virginia Tech, came to the lab, and she was a synthetic chemist. And so she asked the question, are there more microbial bile acids? And really, how do we find them? And so she had this idea that to be able to get our hands on the MSMS spectra of these bile acids, that she would do combinatorial synthesis and then run LC-MS on this, so that we have these MSMS spectra of known structures, and then use something called reverse metabolomics, in that we first have the compounds and then we search if these compounds are actually observed in public data. And the way she did that was, we had just introduced this tool called MAST. MAST allows you to search with this MSMS spectra. It allows you to search the entire public domain, untargeted metabolomics data, to find the files that have those MSMS spectra. Now, where does the data come from that we search? And that comes from the GMPS ecosystem that we introduced in 2016. This is a resource that's accessed probably 450,000 times a month now. And basically what this is, it's both a data repository as well as a knowledge capture system. So if you have any MSMS spectra of known molecules, you can capture this information. If you have a known molecule in your data, you can annotate this. If there's any metadata associated with it, you can add this information as well. So you get this community knowledge that builds up. And this has grown to 3,000 public projects, about 700,000 LC-MS runs, and about 1.2 billion MSMS spectra. So in other words, Emily is searching 1.2 billion MSMS spectra, and you have to realize that there's a lot of computational needs and an infrastructure that needed to be developed to be able to search at that scale. And so when she does a mass search, she finds these data sets. But then within the GMPS ecosystem, the metadata can be curated with a redo system, and about 10% of the data that's currently available is curated in this fashion. But this has controlled metadata vocabulary associated with phenotypes, species, and sample types. And that's really important, because now, as soon as you see a spectra, you have this link to the public data, and then you can begin to connect it to phenotype, species, and such. So I'm only going to show you the bile acid examples, but you have to realize that we also did this for lipids and some other lipids. So she synthesized 176 of these bile acid amidates. So whenever there's an amide form here, Alan Hoffman called these amidates. So I'd like to continue to honor him in that fashion. And so when we search the public data, these are our matches we get, where on the bottom is our reference library that we created, and then on top are examples of public data that match. And so because we have these, we have this controlled vocabulary, we can start to explore how it's distributed. So you can look at, let's say, biofluids or tissue in which these have been detected. So many of these bile acids we see in the feces and the cecum, but you can see them pretty much all over the place, in the urine, upper digestive tract, oral cavity, liver, etc. We also have information on some disease states. And so we can start to look at this in terms of, here on the bottom are the modifications shown. And then we look to see how these modified bile acids are distributed among these diseases or disease states or health conditions. And one of the things that really stood out is that we had a lot of spectral matches in IBD data sets. And so this afforded us to build a hypothesis, maybe they are associated with IBD. And so now we've decided, okay, we're going to go deeper into those data sets and look at the peak areas associated with these different bile acids. And notice that we call these dihydroxylated bile acids trihydroxylated bile acids, because at this stage we do not know the regiochemistry or stereochemistry. And so when we do peak area extraction, we can clearly see that we have these asparagine dihydroxylated bile acids. They tend to be increased in the amounts, or at least the peak area that we observed in Crohn's disease in relationship to non-IBD. And we found quite a number of these that tended to be associated with Crohn's disease in relationship to non-IBD controls. And then there's a pediatric IBD cohort that, if you look at the host-derived taurine conjugated bile acids, up on the antibiotic treatment, you see them increase. And whereas these new bile acids, they tend to decrease upon antibiotic treatment. And this is consistent that they're likely made by microbes. But now we wanted to validate our observations that they are IBD associated. So we sent our synthetic standard to the Broad Institute, because they had published this wonderful paper with a pretty decent cohort of where the N is much larger than what we had. But now we were able to do retention time matching as well. So we're not talking just about trihydroxylated bile acids. No, we can name the specific bile acids to which they're attached to. So here we have alanine cholic acid, alanine chenodeoxycholic acid, etc. There's a couple of them that we could not distinguish, so the UDCA versus HDCA with the glutamine attached, but most of them we could distinguish based on retention time. And what you will see is all the ones with stars are statistically significantly increased in Crohn's disease in relationship to the non-IBD controls. And then we did a study with the Bowen lab at UC San Diego. She had this cohort where they have flare-ups, basically. So do they have active current symptoms? And again, we tended to see these bile acids increase in the amount when they had these active flare-ups. And so clearly these bile acids are connected to Crohn's disease in relationship to non-IBD controls. And so the question comes up is, can these microbes produce these different bile acids as well? And so we've shown this originally for these 3 bile acids. And there were hints based on public data. When we remined this public data, so this was an age cohort from 0 to 200, we see a lot of these new bile acids emerge as the microbiome starts to come in. The other thing that we see is after an antibiotic treatment in animals, you see a lot of, almost all of these will disappear. And then many weeks after antibiotic treatments, you start to see these new ones emerge. And then if you do a fecal transplant in humans, you can see a complete shift in these bile acids. And so all of this is hinting that the microbiome is involved. And so we cultured about 200 gut-derived microbes anaerobically, and you can see that they belong to specific classes, or at least the producers belong to specific classes, such as Actinobacteria, Clostridia, Bacilli, and Fusobacterium. So the question that we got also during the review of this paper is essentially, why have these bile acids never been seen before? And I think this is really a wonderful example of that the scientific community looks under the light post and rarely moves outside of it. But once this light post has been moved, lots of people are starting to see them. In this COVID data set, here they've shown bacteriophyla to transform these bile acids. Here they had an ingestible pill, and they see these bile acids in the digestive tract. And this is a similar type of paper. And then here they recently showed that a microbial catalyzed conjugation of GABA and tyramine to bile acids. But then, of course, we often want to know, do they have any functions? And reality is yes, but there's a lot that remains to be explored. So we did some T-cell assays differentiation, because that sort of differentiation into different immune cells is relevant in IBD. And so we looked for IL-17 and interferon gamma, as well as IL-10. And you can start to see that different bile acids are able to differentiate into different sort of T-cell differentiation or immune cell differentiation. But if you start to now look into the literature, the original one, we showed that they had some effects on FXR. This paper shows FXR and then downstream TGR5. In this paper, it's shown that they affect germination in C. difficile. Here, there's an association with brain injury in premature infants. This paper shows PXR, TGR5, stem cell differentiation, alteration of the leaky gut. This showed a whole panel of different receptors, and there's many more of these this kind of information that is being learned as we speak. But we hypothesized that this was not a really exhaustive search, and so the question that we had is: are there more? How can we find most, if not all, detected bile acids? And so Helena, Psida, and Yasin came up with this really fun strategy of leveraging something called the MassQL. This is really a pioneering approach that Ming Wang developed in the last couple of months while he was a postdoc in my laboratory, but he's now an independent investigator at UC Riverside. But essentially, MassQL is a data filtering strategy. And it really relies on mass spectrometry data. And you have to think about mass spectrometry data, and many of you are using mass spec, some of you might use NMR, but there's only 5 characteristics that defines all mass spectrometry experiments. And you can then create a query language to filter with those 5 characteristics of any mass spectrometry experiment. It doesn't matter if you're doing proteomics, glycomics, lipidomics, metabolomics, imaging mass spec. All of mass spec is defined by those 5 characteristics, and those are the precursor mass and their isotopes. If you do fragmentation, you can look for the fragmentions. If you can look for the delta masses in fragmentation data. If you do chromatography, you have retention time and peak shape, and then if you do ion mobility, you have drift time information. Those are the 5 characteristics that defines all of mass spectrometry. And so you should be able to come up with a simple language to filter for any piece of mass spec information that you wish. And so here's an example for the bile acids that we now create with the MassQL language, but you can do this for almost any kind of molecule, any class of molecule. If you're interested in iron binding molecules, you can look for the iron signatures, chlorine signatures, etc. But basically, if you're mass spectrometrists, you can probably read what this is telling you. But we also have a translator in 9 different languages. And so if we do a query translation, what this is basically telling us, we want all the MSMS spectra that fulfill the following criteria: that it has these 2 ions that we see in the MSMS spectra that really belongs, in this case, to trihydroxylated bile acids. And then we want to fulfill this equation where if these 2 ions are observed, that we then have the precursor mass of the observed spectra that we retrieve, and then we remove the unmodified mass of the trihydroxylated bile acid, and that intensity should show up over on the left side. And so we only take all MSMS spectra that fulfill these criteria. And we created this for the non-hydroxy, which is really not a bile acid, but you'll have to forgive me. I'm going to collectively call these bile acids. And then monohydroxy, dihydroxy, tri, all the way to pentahydroxylated bile acids. And so when we look at the results, we basically start with 1.2 billion MSMS spectra. About 600,000 we were able to filter to belong to these bile acids that we looked for. And then if we look at the number of unique MSMS spectra from those 600,000, we have about 20,000 unique MSMS spectra. And then when we calculate these delta masses, the way I described, where we have a precursor ion minus the free bile acids, and we get a delta mass, we get about 5,500 delta masses. And these are candidate modifications. And so because we have this redo information, we can also start to look at where these molecules are observed. And so the color coding here is based on the hydroxylation state. The number is the number of modifications. And what you will see is pretty remarkable, because generally people talk about bile acids as a part of the enterohepatic system, meaning it's only in the liver, the bloodstream, and maybe the digestive tract, and that you sort of have this circular pattern. But what you will see here is that many distant organs have a lot of bile acids in them: the brain, the lungs, the thymus, etc., the ovary. And we see that not only in rodents, we can recapitulate this in humans as well, because there's a decent amount of human data that's emerging in the public domain. So the next question we had is, can microbes make these as well? And so for anybody who has ever grown 200 microbes, like Pedro and Emily did in the previous study, in the reverse metabolomics discussion, that took a half a year of work for us to be able to get confirmed that these were made by microbes. And so we wanted to come up with a more effective strategy so we don't have to culture these microbes over and over. And so we've started to develop these domain-specific masks, so that as soon as you do a mass search, you can learn, is it found in food? Is it maybe matched to a drug? Or is it found in microbes? And the way we created microbe masks, what we did is we put out a call on social media that if you have data in GMPS of microbes grown in culture, let us know, because then we can curate this taxonomically and link the file to the taxa that are responsible for the production. And we got a lot of reception from the community, and this is outdated. The paper had more than 100 authors on it from I think 17 different countries in the end. And we basically curated 100 million MSMS spectra and linked them to their bacterial, fungal, and production. And so if we now look at the taxonomic tree, we have about 2,300 unique taxonomic names. We then blasted these, creating about 1,800 lineages. And so our final taxonomic tree created 2,800 nodes. And these contain bacteria, archaea, as well as a lot of fungi. So about 12,000 fungi, about 48,000 bacteria, and really generally pretty small number of archaea. And we'd love to expand this a little bit. And then we combined this with the fast search algorithm. So we started to index every MSMS spectra so that you can search as efficient as a text search. And this was really critical because you want to be able to search 100 million spectra. And so now when you search with an MSMS spectra, in this case, the phenylalanine conjugate, you can quickly identify the microbes that are able to produce these in culture. And this is really useful because when we ask the question again, can microbes make these as well? We can search all the 21,000 unique MSMS spectra and then summarize this information in this map right here, where on the bottom you have the delta mass. And notice some of these have some annotations where we know what the delta mass is. So for example, tryptophan, tyrosine, phenylalanine, etc. And then on the left, we have the genera indicated, and on the right is the class level of the taxonomy. And what you can see now is that gamma proteobacteria, Clostridia, Bacilli, Actinomycetia are really prolific producers of a lot of these bile acids. But when we do a mass search, we also find other interesting public data that can be reused to support the hypothesis that these are made by microbes. One of those is from this 2017 paper, where you have these mice treated with a cocktail of 5 different antibiotics. And what you will see is, in the absence of antibiotics, you'll see a lot of these bile acids with these modifications. But now, when you treat with antibiotics, they all disappear, except for this one. And the 107 is the taurine conjugate. Notice that the glycine conjugate actually disappears upon antibiotic treatment. And my hypothesis is actually that the majority of glycine conjugates that we see in human biology or animal biology is really microbial derived. But this means that one of the things that we've recently shown in a separate paper is that taurine conjugates are converted to these new conjugates by an enzyme called bile salt hydrolase. Notice that we still have many delta masses here that we cannot explain. Our hypothesis was, based on the annotations that we did know, that many of these were likely going to be primary metabolites. And so we said, okay, if that hypothesis is true, then we should be able to take the CAC metabolic pathways or similar metabolic pathways or HMDB and try to annotate these delta masses based on the precursor mass or the mass of the molecules that are in those databases and match them up to these delta masses. And this was a really interesting exercise because a series of different pathways showed up. A key pathway that really showed up that appeared to be conjugated to bile acids were these polyamines. And so we decided to synthesize these polyamines and see if they were if they were indeed correctly annotated. And since the data sets to which they mass were deposited in 2017 or 2020, we emailed authors to see if they had these samples in the freezers. Unfortunately, the Turk lab and the Mizrahi lab, they had them available. So that they sent them those samples to us, and we were able to do retention time matching under 2 different chromatography conditions, as well as MSMS matching. And so we were able to confirm 13 of these polyamines, and then 5 of the polyamines, the MSMS matched quite well, but the retention times did not, meaning that they are probably isomers. And so there's a lot more to be learned. And so next we asked the questions, are they present in humans? Since we have the standards available, we could do retention time mapping. And in this case, we looked at a diet intervention study. The Patterson lab collected this data. But this dietary intervention study had an American diet and then 3 different Mediterranean diets or Mediterranean diet with different amounts of meat. And this was really a randomized crossover study. So one person may be having the American diet in the first month, and then another person may be having that diet in the 4th month. But what you will see is that many of these bile acid conjugates, including some of the polyamines, tended to be increased in the American diet and then decreased upon when you go to a Mediterranean diet. Our current hypotheses, also based on some omnivore and herbivore studies that we matched up to, is that this decrease is due to the consumption of fruits and vegetables. But what I think we have uncovered here is this sort of highway where the microbes are doing encoding, and then distant organs are doing the decoding of this information. And the question is, can we get additional support for this hypothesis? If you look at imaging mass spectrometry of a whole animal, for example, so here on the left side, you see the eyes and the brain, and then on the right side is the tail, you can start to see the distribution of a lot of these bile acids. So a lot of classical ones, such as tower hydroxylated or unconjugated hydroxylated bile acids, they tend to be associated with the intestinal tract. But whereas the leucine, isoleucine, we see evidence for those to be in the muscle. Whereas the citrulline, proline, and ornithine, they tend to be localized into the brain, and sometimes really highly specifically localized to really sections of the brain. There's additional evidence when you start to look at the Human Protein Atlas, an expression database. When you search for bile acid associated genes and look at their distribution, what was really striking to me is that both the protein annotations as well as the transcript annotations, that distant organs such as the brain have more bile acid associated genes than the intestinal tract. And so this really shows that bile acids are going to be some of these key regulatory molecules that are really controlled by the microbiome. Now, what messages they're sending, we're going to have to tease apart. Probably we will need thousands of studies that will have to tease apart what those messages are really telling that the microbiome are informing the rest of the human biology. And so, although we have this sort of hypothesis, and it's clear, we believe that bile acids are intimately involved in these messages, but they're not going to be the only molecules that are involved. And so, are there potentially other molecules that are part of this information highway? One of the classes of molecules are these N-acyl lipids that we were interested in, and part of it is because some of these molecules that are made by microbes have already been shown that they do signaling in human biology. In this case, we're showing a GPCR agonist, and this is an N-acyl lipid where you basically have a head group and then some lipid attachment. And so Helena was interested in using MassQL to see if she could find additional of these N-acyl lipids. And so she created these queries for different head groups and then basically looked for these lipids derivatives, as you will. And so this is a representative query that we created. So in this case, we basically want to return all the MSMS spectra that has these lipids from C2 all the way to C30 of these different lengths that you see right here. And then in orange, it defines the head group that we're looking for. So this is just one head group. In total, she searched for 64 different head groups. And so she matched about 10 million MSMS spectra belonging to about 1,400 unique compounds. And what was really striking is that these head groups seems to be changing based on the age. So there seems to be a trend that younger individuals have very different head groups than the older individuals. And then we can again ask the question, are they made by microbes using microbasts? And then we can create this one summary. And so I'm going to carry you through this summary real quick. So on the bottom here, it's indicated with C2 to C30, and basically 0 double bonds. So 2 carbons, 0 double bonds, 30 carbons, 0 double bonds. Here we have the C3, 1 double bond, and all the way to C30 with 1 double bond, and so on. And what you will see is that a lot of these unsaturated lipids are produced. And then as you go to these more saturated ones, they're less frequently detected. And that may simply be because maybe they have hydroxylations attached to them and ketones that we're not capturing or other modifications. But then we also superimposed which ones are observed to be produced by microbes in culture. And you can see many of these are produced by microbes in culture. And then finally, the ones with stars are the ones we've created synthetic standards for at the moment. We're working on creating synthetic standards for all of these, where we then get the MSMS confirmation of the MSMS pattern. In the future, we'll do retention time mapping, but that has not been done yet. So you can see quite a large number of these have now been confirmed with synthetic standards. But I believe that these are also part of the information highway, because we can see some of these are really distributed in the brain, some of these are in the heart, some of these are in breast milk. And so it will be interesting to tease apart what kind of signaling events each of these molecules create. And it's clear that these nasal lipids are involved in signaling. Some of the nasal lipids, such as cannabinoids, are a part of this, belong to these classes of molecules. But I want to leave you with some take-home messages. I think we now have some new and targeted metabolomics tools really to discover microbiome-derived molecules. Please make your data publicly available. The lipidomics community is poor at making their data accessible. It would really be transformational for the field if you made your data public. Once you have a large enough resource that's available, new data science technologies can be developed to really mine that information. I think we're going to be uncovering many new different biochemical pathways, probably at the level that hasn't been seen since the 1960s. So I think this is really the data science capabilities that the metabolomics and the lipidomics community is building now is really going to transform how we're going to look at lipids and metabolites in both animal and human biology. And then finally, of course, I get to speak about this work. These are really the fantastic people. I highlighted some of the key players throughout the talk, but this is the team that made a lot of these resources possible, as well as funding from NIDDK that is allowing us to build a collaborative microbial metabolite center. And then finally, I want to thank you for your attention, and I will answer any questions you might have.
Thank you, Peter. That was phenomenal. Which is great, we have 15 minutes for questions, because there are a lot, I will say, and more
Terrific.
to come, I'm sure. I'm going to take chair's action and ask you the first question, and that is that, I mean, it's phenomenal, the amount of molecules that seem to be coming out of the microbiome and ending up in diverse organs. Is this something we want to happen? Is it good? Is this signaling that's designed and evolved with us to be doing something productive, or do we want to not have this happen? And what are your thoughts on that?
I think we do, well, depending on what circumstance you want this to happen. If you have a leaky gut, there's many of those molecules you don't want to have circulating. And so if you have health issues and your intestinal barrier is not functioning the way it should, then it's problematic. But there's many of these molecules that are really key for human health. And so I think it is important that these molecules exist. They do immune regulation. They define satiety, whether you want to have an appetite or not. So there's a lot of human physiology that these molecules control, and rightfully so, because if you don't have the right nutrients, you probably don't want to have certain signaling pathways activated. Whereas if you have the right nutrients present, then you do want those to be activated.
Yeah.
So I think these are very important.
Yeah. And there's a lot to discover about this. So, yeah, Nee, can you speak?
Yes.
Okay, you go ahead.
I hope we'll be fine.
Yes.
So yeah, really nice, impressive work and lots of data. I have two questions. The first is, is there any further confirmation that all this mass shift, which corresponding to amino acid, is actually the exact amino acid residue on the biacids, or there are some potential isomers?
You have to do retention time matching. So at that point, I mean, it's still possible that this could be a D configuration or L configuration, but it's really the best we can do. If you do this on multiple chromatography conditions, that's what you have. what you could do is you could probably hydrolyze them off and do Murphy's analysis. But given that cholic acid is one of the dominant bile acids that exists, let's say in human biology, you have to have some reasonable confidence that if you do two different chromatography conditions and the retention time remains the same, that's the molecule that has been observed.
Okay.
Thank you. Yeah.
Nata Dudkina, can you unmute yourself now?
Yes.
Go ahead.
Thank you so much for such a wonderful presentation. I guess I was wondering, with a lot of those bile acid conjugates being found in the brain, do you have any hypothesis for, do they act through the vagus nerve, or can they enter the brain directly? Yeah, just wondering about that.
It's a great question. It's completely unknown. What I was surprised about is that the BAAT enzyme that conjugates taurine is also observed in the brain. So I don't know. We do know that in the 1960s, people had shown that bile acids are synthesized de novo in the brain, but we do not know if they can be actively delivered from the guts. So I think in general, there's some supporting evidence for it. So people have seen, for example, lithocolic acid is definitely detected in the brain. And that's a gut-derived microbial bile acid. But sometimes human biology is more complex than we appreciate. Perhaps there is a human enzyme that can create it that hasn't been discovered. So there's that potential. But all the data suggests that there's some way that there's an active delivery of bile acids to the brain.
Thank you.
As well as de novo synthesis.
Thank you. Matt Conroy.
Yeah, thanks, Peter. I might have missed this, but are the microbes making the bile acids de novo, or are they taking host bile acids and then transforming them?
That's a great question. So the bile acid core themselves, so far we haven't found an example where it's directly made by microbes. Now, cholesterol has recently been shown to be made by microbes. So it would not be unreasonable that at some point we're going to discover microbes can make cholic acid or chenodeoxycholic acid from cholesterol, given that they can make cholesterol de novo.
Yeah.
What I show here is just direct coupling of the amines or amino acids to the bile acids cores themselves.
Yeah, thanks.
Okay, so next is Steve Barnes.
Hi, Steve. How are you?
Yeah, I'm fine. Terrific talk as ever. How much do you think that happens in the human in terms of the glycine conjugates? Are the glycine conjugates in bile coming from the liver using the gene that we isolated, or is there a proportion that's coming from the gut? I did some experiments a long time ago with rats. Now, rats, obviously it's similar to, and they have a high amount of taurine conjugates. When we put radio-labeled bile acids into the portal vein, then what came out in the bile was a mixture of glycine and taurine, roughly 1-to-1. When we put doubly labeled bile acids in the gut, the taurine conjugates came back, but the glycine conjugates did not. They exchanged out the glycine. So I'm trying to get a sense of, for the human, how much of the glycine conjugate in bile and the rest of the intestine is coming from the bacteria.
I think this is so it's somewhat undetermined. In the mice, it seems to be pretty much all glycine comes from bacteria, because as soon as you deplete the microbiome, almost all the glycine disappears. There may be a small basal level of glycine conjugates. I don't have a good answer for you in the humans. What we do see is for the taurine that it significantly increases upon antibiotic treatments in humans, and that's consistent across many different studies. There's some evidence that the glycine significantly decreases, but you're also never fully depleting the microbiome in humans under those circumstances.
Mm-hmm.
My guess is that the majority of the glycine conjugate is derived from the microbiome. That's my hypothesis, and partly it's some of these early colonizers. So Bifidia, bacteria, Clostridia, not Clostridia, Bacteroides. Those are the ones that are really active glycine producers. So they tend to make a lot of glycine conjugates.
Mm-hmm.
And so that would be consistent with that the majority of the glycine conjugates seen in humans is made by the microbiota.
Yeah, it's about three, the glycine conjugates are about 3 times as much as the taurine conjugates. So that's pretty massive if it were true.
Yes.
Yeah. Okay, thank you.
Okay. Thank you, Steve. Ed, Dennis.
Hi, Peter.
Hi, Ed. How are you?
Good. Just to focus on the lipidomics side for a minute in technique. Lipidomics people usually start with a lipid extraction, but many of your bile acid-based targets are quite polar. Do you routinely extract before the LCMS? And if so, what technique do you use? And principally, you would, in many cases, you have amino acid conjugates, and you'd want to compare what % of the amino acid is conjugated that's free, and questions like that. How do you approach that question?
How do I approach the question? So, I mean, lipidomics community tend to indeed do a lot more non-polar extractions. So the public data does contain some lipidomic data that people have deposited. So we will research that data as well. In my own laboratory, we tend to do more polar extractions, really sort of in methanol or ethanol extractions. And maybe that's why we're seeing a lot of these conjugated bile acids that previously were not observed. We also tend to look in positive modes as opposed to negative modes. And that may also be why we see a lot of these N-acyllipids, as well as the N-acylated bile acids. So yeah, that's probably the dominant extraction we use is sort of a methanol or ethanol extraction.
And so, in a way, you're not seeing as much of the traditional lipids as the traditional lipidomics community sees, and so it's weighted heavily on a side that we don't normally look for, or probably miss in the extractions. Possible.
So I mean, in terms of the standard bile acids, the trihydroxylated, dihydroxylated bile acids, the free acids, they're pretty soluble in methanol and ethanol. So I don't see there to be a limitation from that standpoint. But if you look at a lot of the more very hydrophobic ones, let's say, hexanoic acid or hexanoic acids, those may be less extractable under those conditions.
So like the cholesterol esters that we would traditionally see, you may not see the cholesterol esters in the other less polar backbone.
Maybe not as intense. I mean, for 2 reasons. We often look in positive modes, and so they don't ionize as well in positive modes. We will see them if they are in very large quantities.
So, Jason Rodensal?
Yes, hello, thank you. So I have a somewhat naive question of, let's say, one is doing some untargeted mass spectrometry. You get some MZ values you're interested in, you suspect it may be a polyamine or biolipid, or excuse me, bile acid, N-acylipid, etc. I feel that your work has essentially de-orphaned so many different MZ values and MSMS spectra. Is there something we could do similar to searching lipid blast or lipid maps to probe your data?
Yeah, so all this, the GMPS ecosystem is public. So all the reference libraries are in there. You can also publicly download them. There's a libraries tab in gmps.ucsd.edu. You can do mass searches yourself. If you get a match, it also automatically, if there's a reference library match, you will get that as well. So all this is publicly accessible. And if you can't find it, let me know, because yeah, I will direct you to the access to it. There's also extensive documentation of how to use it. So molecular networking, feature-based molecular networking, propagation of annotations, and so on.
Mm-hmm.
Fantastic. Thank you.
Okay. Amir Ravandi.
Great. That was a cool talk. My quick question is, I think you showed the power of having sort of fragmented data. A lot of the lipidomics community still rely on MRM-based, like quadrupole analysis. For untargeted approaches to lipidomics, should we lean more towards this data-independent analysis so we can potentially annotate or find undetectable lipids later on?
It depends on what your goal is. Is your goal, is it quantitation, and for specific molecules of interest? So then you probably want to do targeted. Untargeted will never be as quantitative as sort of the targeted approach. Targeted approach is also more sensitive. If you want to do discovery, I would start with untargeted sort of LCM SMS based approaches. So it really depends on what your goal is, your end goal of your particular study. Now, the ideal scenario is that the untargeted discovery leads to the discovery of these molecules that we then create, the community create standards that are made accessible so that you can do targeted quantitation. So I would not advocate for one or the other. It depends on what your goal is. It's very similar in sequencing. You have qPCR if you want to do quantitation. You can do metagenomics if you want to sort of do discovery science.
Okay, thank you. So next question to Al Merrill.
Since you've been finding so many amine conjugates, I was wondering if one of the amine conjugates you've come across is anything connected to one of the sphingoid base backbones, mammalian or some of the many bacterial, and of course there's a lot from eukaryotic microorganisms like yeast.
We have simply not looked for them. So in principle, you could probably create your own query for this and see if you can find it.
Thanks.
Okay, so Alice.
Thank you for his amazing talk. It was really inspirative, amazing. I have one question. Just as you are using a lot of public data, public repositories and databases, how do you ensure the curation of the data?
So I get to ask this question a lot, and so there's a couple of ways that we can answer this. One is the raw data that's deposited. So those are studies that are deposited. Even the best study has poor data in it. So there will always be signals at the lowest level of detection. However, when you search it, your search criteria defines the quality of information that you're willing to retrieve. And so it's really to the end user. And so it's not so much that the data that's deposited has a quality curation, but it's how the data is used that has the quality built into it. And you have to be willing, are you going to be greedy, meaning that you're likely going to capture more false and incorrect pieces of information, or you're going to really be rigorous and focus and minimize errors that it would retrieve. Now, in terms of public annotations that are being made, there are three scenarios that we have. We have the gold libraries that are deposited, the silver and the bronze libraries. The gold ones are really restricted by a very select number of people can upload gold libraries. But those are the synthetic standards that people have run, and those then get uploaded. Those are heavily curated. The silver ones are the published ones where people have NMR confirmation of the molecule, and they've uploaded, and you have a reference for it, whereas the bronze, anything goes. And so you can decide when you search the ecosystem what level you want to utilize. But my experience is that I've never really seen people only selecting the gold library because they want to be able to see and discover what the molecules are. We do have something called living data, where we then curate based on the star rating the quality of matches that we get. And this is 1 star is basically incorrect. 2 star, it's likely an isomer. 3 or some family member. And then you go to 4 stars where you know that it's correct based on the biological system you're studying, based on retention time matching, etc.
Okay.
Okay, thanks a lot.
Okay, and I think because we're over time now, we'll do one last question. I'm very interested in the presence of previously unknown bile acid conjugates in various organs. 1, do you also see the presence of unconjugated bile acids in extrahepatic and intestinal organs? 2, are these conjugated in organs such as the brain by local microbiome? 3, or are they produced by the gut and then mobilizing to extra-intestinal organs? Big question.
Great questions. We definitely see unconjugated bile acids. Just as a lot of the modifications may actually be unconjugated bile acids that we're looking at. So there's a debate whether the brain has a local microbiome. So I think that's a scientific question that's still undetermined. However, if you look at host enzymes, the BAAT enzyme is expressed in the brain. And so the BAAT is the one that is responsible for coupling taurocholic acid to the free acids. So in principle, that should happen there as well. Also, in the past, it was described that it also conjugates glycine, which is based on, but I think we need to double check if that's indeed accurate still. But at least the BAAT enzyme is there. So likely there will be conjugation that happens directly in the brain, but by the host cells. The microbiota, it's still debate whether that exists in the brain. And I think the question of are they being produced in the gut by the gut microbiome and then mobilized to extra-intestinal organs? Yes, that is definitely happening. Whether the brain, that happens in the brain could be a debate still. There is clearly lithicholic acid that has been detected in the brain and other microbial bile acids, the free acids. So I presume there is a linkage there, but nobody has actually demonstrated directly of bile acids from the gut going to the brain. So that will need some feeding studies and such to see if that indeed takes place. If that's not the case, then there has to be some sort of local production by microbes or maybe the hosts that exists.
Yeah, it's going to be really interesting to work this out, isn't it? Yeah, fascinating work. Yeah. But thank you very much, Peter. That was excellent. Really, really great talk. And I look forward to seeing everybody or whoever is able to join us next month for our next webinar. Thank you.
Yeah.
Thanks, everybody.
And if anybody has questions, they're welcome to email me.
Absolutely. Bye now.